Yeah, so like, yeah, I got it.
Hello.
Better for sure, but I want to take the tram because I don't want to be cold because I took just like all kind of integrators and vitamin C.
It's the
But I'm
best.
coming late after the.
Okay, cool.
Me too.
I have everything prepared, but I will be late in 5 minutes, so I connecting from here.
Sounds good.
Hello.
Hello, hello,
One
hello.
of you has the I look good, I'm happy filter, and the other one has the robotic filter this morning.
Yeah, I have a back pain and a kind of torticollis, so it's not good. So I will stay like that.
Too much
It's been a
meditation,
rough
man.
weekend.
Too much meditation.
Not rough at all. Yeah, I need meditation.
Gotta take a break. You hurt your neck.
Right.
Alright.
Thanks for joining us this morning. It's actually 9:30, so let's make a start. Super exciting today. We have a review from Science from 2019, I believe, that talks about the gut-brain axis, which was a question that came up in our previous sessions, and none of us really knew how to answer the question. And today we have the expert, Marie Falkenberg, who's doing her PhD on the topic, and she will present the paper to us. Marie, take it away.
Yeah, let me share my screen.
For everyone else, I shared the paper and the link in the chat already, and as always, questions afterwards. Same for the people over on YouTube.
Can you see my screen?
Perfect.
Cool. So yeah, as Steffi mentioned before, it's a science paper on microbiota in the social brain. And in this review, the authors look at social behavior, so how we as humans, but also animals, interact in social situations, so when we are in a group. And to begin with, let me show you this one here. So yeah, social behavior is any behavior that we have when we are in a group or if animals are in a group. And in neuroimaging studies, it was found that different subcortical regions, which you can see here, are underlying these social behaviors, such as the prefrontal cortex, or the ACC, the hippocampus, the amygdala. Yeah, and we should know that the microbiota, so the bacteria that we can find in our gut, for instance, they may influence our brain physiology and behavior. And there are different pathways by which they can do so, which you can appreciate in this nice picture here. And I'm gonna go over this in a rather simple way so that everyone can understand it, even if you're not familiar with the gut. So there are different pathways by which the microbiota, so the bacteria, and the brain can communicate. And the first one being the vagus nerve, which you can see here, this one. It is a nerve, which is it's a neural pathway connecting the gastrointestinal tract, which is here, down here, and the brain up here. So like let me see, the nerve fibers projecting to regions in the neocortex. And they can interact with the gut microbiota via synaptic connections with endocrinic cells in the gut, so down here. But there are also different pathways, and then there are metabolites, and they can interact by producing neurotransmitters and fatty acids, so like the free fatty acids and the short chain fatty acids. And that is a way they can influence actually the brain physiology and thereby also behavior. And another system for interaction is the immune system. And bacteria can release immune agonists in the blood, and then they can access our brain. And finally, a system that I wasn't familiar with is the olfactory system, so how we perceive smell. And there's growing evidence, at least in the animal kingdom, that there's an association between the microbiota and the brain through olfaction. And all these pathways, this communication between brain and gut is thought to be bidirectional, with social interaction also being able to influence the transmission of the microbiome. Let me go down a bit. Yeah, so in animals, for example, in invertebrate species, they consume each other's feces, for example, and that way it's a horizontal transmission of the microbiome. In humans, it would be, for instance, if we are kissing or interacting with others, we get their bacteria in, and that is able to change our microbiome. Another factor that is influencing the microbiomes of bacteria is the geographic location. So even different species have a similar microbiome depending on where they are located. So the area that we live or the animals live in. Coming back to the social influences. So it was found in germ-free mice, for instance. So mice that don't have a microbiome at all, they were kept like without any, so they had no microbiome because they were kept germ-free. And here we can see that in the absence of a microbiota, they show deficits in social recognition and social cognition. And similar was found in rodents when they were given antibiotics. Antibiotics are not good for the microbiome. They kill a lot of bacteria. They showed deficits in social behavior. And given these interactions, it's often surprising that in social disorders such as autism spectrum disorder and social anxiety, let me see where it was. Yeah, there's evidence that the microbiome is different than in neurotypical or healthy adults. And several clinical studies showed that there's like perturbations in the microbiome. But this needs to be understood a bit more because, for example, in individuals with autism spectrum disorder, sometimes they also have a different diet, which is also a factor that can influence the microbiome. So, and what is actually quite neat is that we can target the microbiota. So we can change the composition of our gut microbiome by giving, for instance, probiotics. And that is an easy way of changing what we have, making the abundance of certain bacteria higher. And it was found that this can have beneficial effects. I mean, this is still like work in progress, but in autism spectrum disorders that on the social behaviors, this had a positive influence administering probiotics. And it's like an easy way to do it. Similarly, diet can change our microbiome, and so can prebiotics. And this might in the future be a way of targeting the microbiome and thereby targeting social behavior. This was it like very quickly. I'm happy to go like more into detail if you have any questions, but I thought I kept it broad so that everyone can get something out of that, but happy to talk more about it.
Lovely, thank you. Very interesting review. And we have our first question already, Michel.
Hey, very, very interesting, very nice presentation. So a first question that come to my mind was this, is we show a lot of significant association between different parameters in our job, and those significant associations are often related to how large is the sample size. So if we have a very large sample size, we can show a significant association. But that opened up something that I don't know if it's in the review or if you've seen it, thought about it. There is an association between the gut system and the social behavior. But how big is that association? How much like the gut influence the social behavior? Is that just a little bit and we show that there is some effect, or is it like really a big effect?
I cannot give you numbers, but I think it's just one of the effects. It's not like the main factor that is going to influence or like be like the defining factor. It's just one of the factors that should be taken into account, but I cannot give you a number.
Okay, thanks.
I have another question from Leah.
Yes. So maybe not more, this question is not related to the social influence, but I was wondering if allergies, like it's not like because, for example, I have nickel allergies, if so kind of allergies, lactose, nickel, so that can maybe there is an interaction studies with the brain. I know that it can give you like migraine or trigger some, is it in this context or is out of the field?
To be honest, I don't know. I know that if you don't eat certain kinds of foods, so for example, if you don't eat anything that has milk in it, you would have like certain strains of bacteria, less of certain strains of bacteria. But for your nickel allergy or something, I really couldn't tell.
No, in general, it's some allergies can like imbalance this system. But yeah, I didn't see that in the when you were scoring the review, so maybe it's out of topic. I don't know.
No, I think it's not part of the review, but I think it's still in topic. But I'm not familiar with it.
Okay, thank you.
Hervé, sorry, I saw you too late. Take it away.
No, I didn't find the raised hand button, so I found it. So yes, thank you, Mary, for this paper. So it's strange. Last week, I saw a discussion, but I don't remember where, talking about microbiota, social behavior, and autism. And this discussion was saying, yeah, there is in autism spectrum disorder in patients, they have different microbiota, and so there's discussed that can be a link between autism and so the link between brain and this disorder. And but at the end, they discussed that also patients have a different diet which affect the microbiota and could be not related to the link between brain and microbiota. So I don't know if you have saw this discussion. I don't remember where.
Yeah, that's one of the main factors, because in autism they have sometimes they have like a very restrictive kind of way of eating, so they only eat certain types of foods. So yeah, that's a big downside. It can still be seen that the microbiome in other pathologies that come along with social deficits, such as social anxiety, is also altered.
Mm-hmm.
But yeah, it's difficult to differentiate. Like, what is the causal relationship between these two?
Exactly. Thank you.
You're welcome.
Cool. I have two questions for you. So the first one is about the rodents with no microbiome. I didn't know that was possible. Is that actually a thing?
That is a thing, yes. You can even, so you have germ-free mice. So you keep them away from everything, so they are not in contact with any bacteria, fungi, or whatever. So they grow up germ-free. And then you can infuse them with the microbiome of someone else, for instance. And that, for instance, was shown to be like, if you have like a germ-free mouse that is like lean, you infuse it with the microbiome of an obese mouse, it will get fatter.
And they just never develop the microbiome?
Until they are in touch with something, yeah.
Okay. And then the second question I had for you was about the olfactory route. So we know that olfaction is very important for social interaction.
Mm-hmm.
But do we already know the mechanism of how that then triggers a cascade to involve the gut, or is that still quite a new topic?
To me, it was completely new. And in that review, they only talk about animals and only certain kind of animals. So I think we don't know much about it yet.
Thank you. Leah has another question.
Yes, another thing that I was curious about. So I'm really interested in the vagus nerve and also the sympathetic parasympathetic balance. And I know that like in the clinics, there is this kind of test, tilt test, where they see if you have overstimulation of the vagus nerve, but it can give a false positive or false negative. My experience is not super reliable, so I was wondering if it's something that you can really measure and quantify if you have this kind of imbalance that then can maybe influence in this gut-brain also interaction, or is far away also. I'm just wondering about my knowledge. And this.
I have no idea.
Okay.
Interesting question, though. That's cool.
Because yeah, there is a way of this measure, but yeah, it's not so solid.
Yeah, I was wondering, how could you measure that?
There is this tilt test, so they see also it's more in the you can say autosomal, like vertigo, if you have this overstimulation, you know, like males, they can also feel it like here, this nerve, vagus nerve stimulation. But yeah.
Interesting.
Just wondering, thanks.
And Chris is joining us with a question.
Hey, thank you for the presentation. I have a question about the time scale of these interactions. So how fast is the gut able to the gut microbiota able to interact with the brain? I mean, like the brain interacts pretty quick, but is the other way in a similar, probably not as fast, but maybe in a similar time scale? Because also I'm wondering on the long term, if you change your microbiota, will it change your brain in more than a chemical way, if you will develop less or more connections on the long term because you change your microbiota?
Okay, so what it says in the review, for instance, that so if you give antibiotics, it kills your bacteria in the gut. Not all of them, but certain strains. And for instance, in rodents, it was found that this has, like, if you give them to them as babies, they still have like effects until their adult life, the rodents. I don't think it's no, it is not that long in humans if you give them antibiotics. But in intervention studies, it's usually a couple of weeks until the microbiome is changed so that you can see effects on the brain. So it's not something that is happening. You give one pill and you see effects.
All right. But if you keep the same, so if you have a disorder that changes on the long term your microbiota, will you have a long-term effect on the brain? You will keep the effects, right?
I would think so, yes.
Yeah. Thanks.
Leah's on fire today.
No, this topic I really like. It's my personal interest. So another question, but maybe you don't, I don't know. If, like, you know, the microbiota, you can balance that with certain yogurts. Is something that is considered in this review, or no? Like, to create the balance.
They're not talking about yogurts, but they're talking about some strains of bacteria, which are, for example, lactobacillus, which you can find in yogurts. Okay. So, yeah, I mean, you can, if you eat certain kinds of food, so diet does influence your microbiome, what you eat does influence, so also the yogurts influence your microbiome. Yeah.
Okay, thanks.
This is putting my clinical hat back on now. There's also a couple of investigations ongoing, as far as I know, where they try to kind of move the microbiome from one person to the other. As you mentioned with the mice earlier, if you have a lean mice and you introduce the microbiome of an obese mice, that lean mouse now becomes fat. And the same logic could be potentially applied for diseases, right? Where you take the microbiome from a healthy person and you give it to someone who has a disease in the hope that they then recover. Do you know anything about those lines of investigations or diseases that are involved in that?
Yeah, I just know that so this is not done for like mild diseases, but like people that have really struggled a lot with their gastrointestinal functioning. In these people it is tried, and it seems to be successful so far, so that they get like the microbiome out of another healthy person, and it can change their microbiome in such a way that they get healthier. I don't know what exact conditions it was, but it's definitely something that is done already. But yeah, not for, it wouldn't be done for anything like, so it's, yeah.
Well, thank you. Are there any other questions? No, everyone's good. Well, fantastic. Thank you so much for presenting, Marie.
Thank you for having me.
Next week, we're going to have Patrick presenting the Arcuate Fasciculus and language origins, disentangling existing conceptions that influence evolutionary accounts. That's a preprint. So that should be quite exciting next week. Do join us every Monday morning at 9:30. And if any one of you or any one of you out there want to bring a paper, just show up, present the paper. Ideally, let me know before so we don't have a clash. But everyone is welcome to join us and present a paper of their interest. Now, with that, I wish you a wonderful and exciting week in science, and I'll see you next Monday. Bye.